Alex Stevens is a research assistant professor and the co-founder of the Advanced Imaging Research Center at Oregon Health and Science University. His research focuses on questions related to neuroplasticity. Alex, thank you so much for taking the time today.
Thanks for having me, Joe.
So, to start, could you talk a little bit about your journey to this research?
Sure. So it really started as an undergrad when I was studying psychology and was interested in questions around human motivation. And in fact, one of the things that really interested me early on were questions around um, psychosomatic illness. Sort of why is it that we get pains in our necks when we're stressed out? um, what's the relationship between the brain and the body? That was something that really fascinated me. And I went on to graduate school and initially was studying issues around uh, human individual differences in personality, things related to social interaction. And as I started exploring that, um, sort of the way we interact and how that affected our physiology, our heart rate, and our pulse rate interested me. How how our emotions and our thoughts were affecting our physiology, and I was working with um, a psychologist named Rebecca Warner who was interested in these social interactions, and uh, so as I worked on that, I became more and more interested in what the brain's role was in those interactions. We saw when people were having a good interaction, if you sat two strangers down, if they actually conversed, that there was some synchrony between their heart rates and their blood pressure would drop. And as I was studying this, questions about, well, what is it about our emotions, our thinking, our um, what happens in our brain, how does that affect our body? And so I decided after I finished my master's to um, actually go directly to the brain. And I joined the lab of Robert Mayer, and who was studying an animal model of memory and amnesia. And that really got me thinking about well, it put me in the brain. And so I focused on looking at how short-term memory was formed and the circuits involved in sustaining short-term memory um, in the frontal cortex and a region of the brain called the thalamus. So I began focusing on that, but was uh, and did my dissertation in that area. um, and as I was thinking about after graduation, after I finished my PhD, I was really interested in taking what I'd learned in neuroscience and combining it with my work in human psychology. Some of the work I'd done in social interactions, and as I was looking for a postdoc after I finished my PhD, I I happened to be at sort of the right time at the right place. This new technique was coming along called functional MRI, which was a way to use a machine, an MRI, a magnetic resonance imaging scanner, to study functional activity in the brain. And the lab of Patricia Goldman-Rakich at Yale was interested in starting some of these studies in the area of schizophrenia. And so I was fortunate enough to go down there, and this was sort of a brand new technique, and I was collaborating with Dr. Goldman Rakich and then John Gore, who at the time was the head of the MR physics in the School of Medicine at Yale. And so I began collaborating with them and working on studies of schizophrenia. And so that sort of got me into the early days of work with human and functional magnetic resonance imaging. And as I spent my time training on that and becoming more and more interested in how the human brain works and how we can use at the time this new technique of fMRI to try to understand how our perceptions, our thoughts, our emotions can be studied with this technique. Uh, I then moved to Oregon, and at the time, Oregon didn't have an imaging center. And so I was using scanners, MR scanners in the hospital here at OHSU on the weekends to do certain studies that I could do with healthy, normal individuals. I wasn't, it was a little too difficult to try to study individuals with schizophrenia. That those are just, they're more complicated studies, and I just didn't have the support at that time. A colleague of mine and I decided to um, that we needed a research dedicated scanner, and so uh, her name is Jerry Janowski. She's no longer at OHSU, but she sort of led the charge, and we put together a plan and developed what's now the Advanced Imaging Research Center at OHSU. And I've been here ever since. My research has evolved over time in some new directions, and the imaging center has evolved over time. So we now have a number of scanners. Some are human-based, so we have what's called a 3 Tesla scanner, and that's about, just to give you an idea of the power, and a 3 Tesla scanner is sort of the standard hospital scanner, but it's got a magnetic field about 30,000 times the strength of the gravitational pull of the Earth. So they're big, strong scanners. We also have a 7 Tesla scanner, which again is even more powerful, that we can use to study the human brain. And then we have um, a couple of scanners that are used in animal studies, um, which are even more powerful. We have what's called a 12 Tesla scanner, which is used to study to do animal studies. And so our centers, got some great equipment and allows us to do some really interesting studies in humans and animal models of illness and all sorts of stuff.
Yeah. Well, I'm super curious about the kind of the capacity of research, and you described this process of getting a dedicated scanner, or
Mhm.
research dedicated scanner. I'm curious what you think about the current capacity for research, and how that has changed over time, and what you see um, kind of the needs or the equipment gaps in research right now, and what that looks like.
Mhm. Um, so the the capacities that we have now in I'll focus on human studies because they're what I know best, and I think that in many ways they're the most relevant um, to human illness and human health. Um, and that's not to say that I don't think animal research is important, because so many of the discoveries that we have started out with animal models. Um, but the technologies that we're able to bring to bear now on research are really stunning, and new techniques and new technologies are coming along every day. I mean, I was at a talk yesterday, in fact, when a new technique of brain stimulation. So this is the ability to affect the human brain non-invasively, has been developed. There's a number of them now, but this was a new one that I hadn't come across before, that allowed the electrical stimulation of deep areas in the brain that until now have required, electrodes being, put into the brain of individuals in order to stimulate those areas. And here I'm talking about primarily Parkinson's disease. So Parkinson's disease is uh, neurodegenerative disease, that second most common neurodegenerative disease in the world now. Um, and it's one of its symptoms is a constant shaking, usually of a limb or the head, sometimes as it progresses, all those things. And it's very debilitating. One of the more effective treatments for it is what's called deep brain stimulation, where you take an electrode and you put it down into an area of the brain called the subthalamic nucleus, which is very tiny structure down in the middle of the brain. And you can then adjust the stimulation to that electrode, and it often provides a great deal of relief to individuals with Parkinson's disease. This new technology wouldn't replace that, but you can actually start testing how well an area may, and this is sort of looking forward a bit, um, but you may be able to now use this technique before you do any operation to see if stimulation in that area would actually have a benefit.
Mm.
So it's really remarkable what people are coming up with is as potential treatments and uh, potential methods for studying the brain.
Yeah.
Um, so as far as um, what technologies out there, there are a lot. I think different centers and universities around the world have certain areas of expertise. I know that all my colleagues, if you ask them, they could all come up with something they wish they had. Um, but I think a lot of the in if we talk about magnetic resonance imaging uh, in particular, there are a ton of things that we can do with it, and people are finding novel ways to use it all the time. And I can tell you a little bit about that if you'd like.
Yeah, that's a good transition. I also wanted to ask you about kind of the mechanics of how they work and um, how they're being applied both in research, but then also how that research is then translating to new ways that doctors and practitioners are doing their work.
Hmm. Yeah, that's a good question. So I'll give you, I'm a psychologist. I'm not a physicist, and these are incredibly complicated machines. I mean, they're pretty remarkable. Um, but the idea is that our bodies and our brains are mostly water, and hydrogen atoms are um, have one oxygen atom, so they're an asymmetric uh, uh, molecule or atom. And when you stimulate them, you put them in a magnetic field. Um, as a whole, the water molecules and the hydrogen atoms kind of align to the magnetic field. Not all of them, but enough of them. And when you stimulate them at a certain frequency with a radio frequency, just like radio waves are broadcast on, if you sort of ping the brain with a radio frequency at just the right frequency, it makes them all sort of spin together. They sort of spin around together. And when you stop the radio frequency, they relax. They all kind of go back. They're not spinning together, they're asymmetric. So, what you can do is you can stimulate the brain with this radio frequency in this magnetic field and manipulate that radio frequency in a way that you can get different signals out of the brain. That's sort of the general physics behind it. And people have come up with astonishing ways to do this, to look at different types of tissue in the brain. So the brain has gray matter, which is where most of the neurons are. It's got white matter, which are the the fibers connecting the neurons, the nerve cells. And there's what's called cerebrospinal fluid, which is just the fluid that's used for bathing the brain and keeping the neurons healthy and removing waste from the brain. And using different techniques, you can highlight one or differentiate the different types of tissue to get anatomical images. Similarly, with the rest of the body, MRI can be used on the entire body. I'm sure lots of people have had an MRI. Um, so we can use it to study the anatomy of the body and the brain. What I use it for primarily is looking at the function of the brain. And so in functional MRI, the primary method we use is something called blood oxygenation level dependent imaging. And we call it BOLD. And so what we're using there is changes in the levels of oxygen versus oxygenated blood versus deoxygenated blood. So the brain relies on oxygen and sugar to run. Those are its sources of energy. And when an area of the brain becomes more active, it's going to need more oxygen primarily and more sugar. So with fMRI, it turns out that the blood is carrying the oxygen. And when the red blood cells bind oxygen, they have less signal. But when the the red blood cells aren't carrying oxygen, if it's stripped off by the area, it it creates a disruption in the signal. So the signal changes depending on how oxygenated the blood is in a particular area. And so we measure the changes in that using the changes in the magnetic properties of the blood as they flow through the little capillary beds and, throughout the brain. And so that's how, when you see those sort of fancy brain maps, with the different colors, there's a lot that goes into that, but it starts by essentially trying to detect changes in the oxygen and deoxygenated blood in the brain. So those are two things. You've got anatomy and you've got the function. We can also use MRI. It's really like a Swiss Army knife. You can do so many things with it. You can also study brain chemistry. So there's a way to look at all sorts of different what we call metabolites. They're proteins and molecules that the brain possesses. And so we can measure levels of those in the brain as well. And so we have all these different techniques. We can study anatomy, function, uh, chemistry of the brain with this one tool, this one machine.
Hmm.
Um, and it's not like you can do it all at one time. And most of these studies, so you can get the full anatomy of the brain in very high resolution, say 1 millimeter cubic um, measures, what we call voxels, in about 8 minutes, 6 to 8 minutes, depending on what you want to do. Functional studies tend to take quite a bit more time because, as you can imagine, the signals we're seeing from the blood are tiny. So we have to average. We have to, do things multiple times and average them together to be able to see the signal. And similarly with what's called spectroscopy, measuring the chemistry of the brain, we have to, be very, we have to sample the brain several times to be able to get a good enough signal. So, so that's sort of a quick overview of how MRIs work and what we can do with them. So
So are the capabilities of the MRI, all those that you just described, are they in every machine, or they have to be tailored? Does every hospital have the capability to do all the tests that you just described?
um, no. So there are most scanners are capable of doing those things. Um, More and more, they're included in the software that controls the scanners. There are sometimes, for certain things, there are important um, pieces of equipment add-ons, what are called coils that go inside the magnet um, to sort of focus the the um, acquisition of the pictures and data. But most scanners can do generally those things. Um, and then there are some things, so for example, studying particular types of chemistry in the brain take very specific types of coils and require a fair amount of expertise to really do. So most scanners are capable of doing these things. It comes down to software and expertise to really pull them off well.
Yeah. Well, I also wanted to ask you about the kind of our societal understanding of the brain and also our understanding of sensory and motor systems. How have they changed over time, and what is our current understanding, and where are we, like, what are the trends right now in how we think about the brain and our sensory and motor systems?
Yeah, that's a good question. The What I'd say is, uh, in my own understanding, I say the longer I study the brain, the less I know,
Hmm.
because the levels of detail that with which we're able to study the brain now has really altered both our understanding in terms of the complexity of the brain, but also the uh, uh, sort of the details and differences among parts of the brain. So when I started in neuroscience, this was 30 years ago, there were about 50 different nerve cells that had been identified. 50. That seemed like a lot. Um, Recently, a study was published that identified over 1,000, and that's probably not complete. So in terms of the complexity at sort of the molecular level, um, it's it's remarkable. And to me, in some ways, I have enormous respect for my colleagues who are able to make sense of a lot of this. So from a neuroscientific perspective, I'd say that we are both getting a handle on the understanding of the systems from, molecules to the whole brain, but at the same time, it's at every step you find that there's actually a step in between. So the real complexities are still being discovered. Um, at the same time, I think one of the things that's remarkable is the progress we've made in treating a number of neurological diseases, that we've developed new techniques using technology to alleviate some of the most difficult uh, uh, neurological illnesses. There's been a lot of progress made. Some of them are being made at the level of genetics, some all the way up to uh, brain-computer interfaces. So our understanding of the brain, um, I would say that it's advanced dramatically. It's changed quite a bit, and there's been some incredible work being done on finding new ways, new treatments, and ways to improve health through a lot of these studies.
Yeah. Uh, Another thing I wanted to talk to you about was the brain's metabolic function. Could you describe how that works and why that's important?
Um, yeah, so I, I'm again, this isn't an area of expertise. I'm doing a little work in this area right now with a team, and I guess that's something I should really emphasize, which I didn't really state before. But science is really a team sport. You know, if you want to do good science, it's often uh, teams of 4 or 5 or 10 or 20 people. And sometimes, you know, now I'll see journal articles that have 50 people all over the world who are contributing. So um, don't take my ignorance as the ignorance of the field. Take it as my own personal ignorance. Um, so, so as as a preamble to your question, metabolism in the brain is like metabolism of the body. So our bodies are made up of cells, and cells they need energy or to function. And so that in the brain comes primarily in the form of oxygen and glucose. And you need oxygen and glucose. They're the energy of the brain. Um, the neurons, what we think of as the important cells that are making memories and making us function, are supported by what are called glial cells. And glial cells are a machinery that actually help produce energy for the neurons. They support the neurons, and more, they're probably playing even more sophisticated roles in that. In fact, they may be involved in memory and learning as well in ways that we haven't come to understand yet. So the brain, um, as far as brain metabolism goes, the brain is about 2% of our body weight. A brain weighs, maybe somewhere around 2 pounds, a little more depending on the person. Um, but it uses about 20% of our energy. So the if you look at the total metabolic activity of an individual, the brain is using 20% of that energy. So it's highly intensive. It's always on. Even when you're sleeping, it's doing things to sort of mop up from the day's activity, but it's still quite active. Um, so uh, in terms of metabolic demand, um, it's highly intensive. It's why if you're um, deprived of oxygen, you pass out. Pretty quickly. The lack of oxygen will affect your brain. If you are um, hungry, if you're in a state of starvation, your brain will actually start cannibalizing itself and use breakdown materials to try to get energy. And so it's a pretty voracious organ, and and at the same time it's highly efficient. It it's functioning all the time. We don't really replace our neurons, so that's been an area of pretty intense study. Um, I have not seen any evidence that convinces me that we actually produce new neurons in our brain. So the neurons that we're born with are the neurons we'll have for the rest of our lives. And so, these tiny little things hang around. And, you know, so that's why it's good to take care of them because they're pretty valuable, and you're not going to replace them. Uh, so um, so that's probably also in part because of these glial cells I was talking about. We need a very robust system to support the the metabolism of the brain and maintain our neurons. Glial cells are reproduced. They can be regenerated. So so I'm not sure I answered your question exactly about the brain's metabolism, but there are it's energy intensive, it's highly efficient, and it is always working. So
Yeah. Well, there's another uh, thing that you mentioned earlier that I wanted to ask you about related to um, kind of the research and what the future of it might be. Are you seeing more applications of AI and machine learning in the work that you're doing, and what does that look like right now?
um, definitely, I mean, AI can be a really great tool. It is helpful in a number of ways, in terms of uh, uh, identifying molecules, for example. It can model uh, almost every single way a molecule can be shaped, for example. It's being used, attempting to be used in screening brain scans, so MRIs. So as a tool to help radiologists perhaps identify um, abnormalities in the brain. Um, in fact, that was thought to be like that was going to be the first frontier that AI was going to crack. It turns out it's not as easy as people thought, but it's becoming a helpful tool by identifying things that may be abnormal, and then radiologists can sort of hone in on those and look more closely at them. But and it's actually used in a more mundane way as well. So the scientific literature is enormous. There are tens of thousands of articles published every year, and it would be impossible for me to read everything, even in my particular areas of interest. Um, I can use AI to look for specific things in the literature to try to find um, articles that discuss a particular issue I might be interested in. And so it's a tool to help me search what's happening in areas I'm interested in. And you always have to be careful because, we've all heard about AI hallucinating. And so you have to then go find those articles it's referring to and make sure they're real and that it's interpreted them correctly. But it's a huge time saver in terms of trying to get a handle on literature as we're working on a particular topic. Um, People are using AI in new ways of trying to understand data, to find patterns in data and find uh, patterns in all sorts of things to come up with new solutions to problems to try. So yeah, AI's showing up and people are finding new ways and really creative ways to use it. So I imagine as people get, and probably, younger scientists are probably using it in more novel ways, um, because they're growing up with it. And so I think there are generational differences to some degree in how they're being used, how it's being used.
So the last question I want to ask you is about the future and or a couple of questions around the future that what do you see as the future of this research and do you see any breakthroughs on the horizon and what kind of outlook do you have for the technology and the work that's going on right now?
Mhm. So I would say if I come back to fMRI, studying functional activity in the brain, I think that it's as I said, it's sort of a well-worn path right now. People are still doing very good work with it. I'd say the future for it may be. I've seen some things that in the last, say, the last decade, and maybe even the last 5 years, fMRI is being used more and more as a way to identify brain regions that may be functioning abnormally in particular disease states. So I'll give you an example. There's an area of the brain in the frontal lobe which seems to be associated with treatment-resistant depression. And a great deal of work has gone into trying to understand the circuit that's involved in depression and its role. There are some studies that have been ongoing for a long time, but more and more people have found that they can use a method of they can use fMRI to identify an area in the brain that seems to be critical in depression. And they use that information to guide what's called um, transcranial magnetic stimulation, which is a means of stimulating the brain without opening the skull. It's a non-invasive technique for um, influencing the brain activity. And this is proving to be extremely effective in treating people with um, major depression who have never responded well to medications. And so I think that's sort of a harbinger of what's to come. Right now, I'm working on a study looking at pain, uh, people who have pain associated with particular type of arthritis. And the idea here is if we can understand the role of the brain in pain, because sometimes the pain seems to be uh, present when there's no sign of inflammation in the tissues. We think what might be happening is the brain circuits involved in pain may be hyperactive. And if we can find somewhere in that circuit that is um, targetable by something like TMS, transcranial magnetic stimulation, we may be able to find a way to quiet down that circuit and relieve people's perception of pain. So I think that there are techniques like that which are going to, we'll see more and more as we get a better handle on specifics of circuitry. And it may be that, many neuropsychological issues, depression, uh, uh, can be treated both with therapies and with things like TMS. And I think fMRI plays a role in that by helping us understand those circuits.
Yeah. Well, Alex, thank you so much for taking the time to talk to me and share your insights and your research. And I hope we can continue the conversation down the road.
I'd be happy to do it. Thanks a lot, Joe.